Okay, 3, 2, 1, it's jam.
What's up, science fam? Why do we even breathe? Why do we need to take in air all the time? It's a hassle when you realize that you have to breathe in and out. Like when you're conscious about it, it's weird. But what exactly is it for? So it's related, of course, to this process where oxygen is a reactant and we get ATP and a bunch of other stuff from it. So this is called cellular respiration, and that's what we're going to talk about in this session. My name is Sermon, and this is Science with Sermon. Let's do this. Cellular respiration. So it's the breakdown of biomolecules to generate chemical energy in the form of adenosine triphosphate, or ATP. So in this session, we're going to discuss the processes, structures, and enzymes involved in it, describe the steps in the different stages of respiration, and describe different types of anaerobic respiration. So basically, food begets energy. But in chemical terms, we can think of it as glucose being converted into ATP. So the organelle responsible for this ATP synthesis is the powerhouse of the cell, the only thing that people ever remember, the mitochondrion. So the mitochondrion has all of these special parts. So we have the matrix, the fluid inside the cell. We have the cristae, the folds of the inner membrane. And of course, if there's an inner membrane, there must be an outer membrane. Now take note, your mitochondrion also has its own DNA, its own ribosome. So there's a theory about the mitochondrion and the chloroplast being free living cells before, but we'll get to that some other time, most likely. So why fold up the inner membrane into cristae? Why have these wrinkles on your inner membrane? That's because, again, more surface area. That's more space for enzymes, meaning more molecules for ATP synthesis. So how do we get stuff into the mitochondrion in the first place? How do we start processing your glucose? So before we get into the mitochondrion, outside the mitochondrion is the squishy stuff of your cell, your squishy liquid. So the cytoplasm is the squishy fluid part of the cell, specifically the cytosol is the fluid part of it, and that's where all of your cellular respiration begins. So the first thing that happens is, of course, we have glucose. So this is the chemical structure of glucose. He's a big boy. He's like a giant Piato's chip, and he won't fit through the mitochondrion membrane, so we have to break your glucose up. So in order to break it up, our first stage of cellular respiration is glycolysis. So literally glucose breakdown, glycolysis. So glycolysis, literally glucose breaking, it's the breakdown of glucose in the cytoplasm of the cell. So even though respiration mostly occurs in the mitochondrion, it begins outside outside the mitochondrion. So the simple version is that the glucose gets broken down into pyruvate or pyruvic acid. So over here. But of course, we're standard, we're hardcore. So this is what we're gonna talk about. Yeah, all of those. So glycolysis has two phases, the energy investing phase and the energy payoff phase. You can't get energy if you don't give something first. Okay? So you get stuff after you give stuff, right? So first, you gotta invest that energy. So two ATPs used to make glucose unstable. This is what's happening over here. So your ATP gives phosphate groups to your glucose and it makes it wobbly and unstable. This unstable form is the fructose 1,6-biphosphate form, or F16BP. So just remember it as F16BP, but again, its full name is fructose 1,6-biphosphate. It'll break down into DHAP and glyceraldehyde 3-phosphate. So that glyceraldehyde 3-phosphate is the same molecule you may have encountered in photosynthesis, your G3P. So again, your glucose is broken down into basically G3P and DHAP, which your DHAP will be converted into G3P as well. But it doesn't stop there. Your G3P is still not an ideal form for it to be shoved into the mitochondrion. So we go to the energy payoff phase. So we put in 2 ATP, but now we're going to take our energy back and get some interest. So what happens is that electrons get taken from G3P by an electron transporter NAD+. So take note, electron transporter here in respiration has no P. It's NAD plus, and then it becomes NADH. One way to differentiate this from the electron transporter in photosynthesis is that for photosynthesis, there's a P. It begins with a P, so it's NADPH in photosynthesis. In respiration, there's no P. Okay, there's no P in respiration. Next, your phosphate gets taken away from G3P by adenosine diphosphate with 2 phosphate groups. It becomes 3 phosphate groups, becoming ATP. So what happens is that for each G3P, you can get 2 phosphate groups. All right, so again, we can get 2 phosphate groups from G3P. Sir, where did the other phosphate come from? There's free-floating phosphate, and the energy of breaking down G3P allows your ADP to get free-floating phosphate groups. So anyway, the point is each G3P can make 2 ATP. And now, how many G3Ps do we have? Again, from your glucose, we broke it down into 2 things. This one was converted into G3P, so we have 2 G3Ps, which each makes 2 ATP. So in total, we get 4 ATP from 1 glucose. So here we see that even though we invested 2 ATP, we got 4 ATP back. So, 4 minus 2, you have a net gain of 2 ATP. So let's focus first on the electrons that were taken by NAD plus there. So again, this is an example of redox reactions. Just to review here, oxidation is the loss of electrons, while reduction is the gaining of electrons. So you can remember it using Leoger or oil rig. Oxidation is loss, reduction is gain, and so on. So what about transferring phosphate? Are there different kinds of reactions for transferring phosphate? We have substrate level phosphorylation and oxidative phosphorylation. These are complicated sounding terms, but basically, substrate level phosphorylation just means the phosphate comes from a molecule, like how we got the phosphate from G3P. We ripped off the phosphate from one molecule and shoved it onto ADP to make ATP. That's substrate level. Oxidative phosphorylation, on the other hand, is phosphate that's attached using energy from the loss of electrons of your electron transporter. So we'll see that later on. So we've only seen substrate level phosphorylation so far. So again, in glycolysis, we started out with 1 glucose and then we invested 2 ATP, but we ended up with 2 pyruvate molecules. Again, at the very end, once you've stripped it of electrons and phosphates, your G3P becomes pyruvate, and we get 4 ATP and 2 NADH from your stripping of your G3P. So your net total is 2 pyruvate, 2 NADH, and 2 ATP. That's just from the first stage. Now, the next stage of your respiration is to prepare your pyruvate for processing inside the mitochondrion. So once it's shuttled into the mitochondrion, we have the transition stage. So the transition stage is when we modify pyruvate to get it ready to get electrons ripped out of it some more through oxidation. So this happens in the mitochondrial matrix, and this is what happens in the transition stage. So again, we have your pyruvate. It is involved with CoAsh or CoA SH. That SH is just sulfur and hydrogen. So what happens is that your cofactor A will replace a carbon from pyruvate. The carbon that was replaced will become carbon dioxide, and in the process of replacing the carbon, there's free electrons taken by NAD+, which becomes NADH. And so your final product is acetyl-CoA. Literally just replaced 1 carbon atom with cofactor A. And this cofactor A makes your acetyl group ready for oxidation. So in your energy and metabolism lecture, you may have remembered the cofactor term. These are enzyme helpers. So that's the point of CoA. It helps the enzymes that are going to come up in the next stage rip off the electrons from the rest of these from the rest of this acetyl group, right? So again, don't forget we have 2 pyruvates that we got from a single glucose. So these 2 pyruvates, each of them will become 1 acetyl-CoA. So we get 2 acetyl-CoA for oxidation and 2 NADH for electron transport. And of course, we have carbon dioxide as waste. So, no breaks here. Next, we have the after the transition stage, we have the Krebs cycle, step number 3. So in the Krebs cycle, we oxidize acetyl-CoA to get a lot of electron transporters. And this also happens in the mitochondrial matrix, the fluid of the mitochondrion. So it's a bit complicated. So this is the simplest thing we can find where it's focusing on the substrates and the products. When we say substrates, again, these are the molecules that are part of a metabolic pathway. And the products are the things that exit that pathway. So these things here are the products, while the things in the middle are your substrates. Sir, there's so many substrates. How are you supposed to memorize them? We have a mnemonic. So the mnemonic is, can I keep selling substances for money, officer? Or can I keep selling whatever S word you want to put there for money, officer? Same mnemonic that med students use to memorize the Krebs cycle. And it stands for citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. Now, some of these substrates might seem familiar if you've watched the photosynthesis lecture video already. Your malate and oxaloacetate are here, and they're also involved in some photosynthetic pathways as reservoirs of carbon. But in this case, it's just part of the Krebs cycle, or the citric acid cycle. So, 2 names for it: Krebs cycle, citric acid cycle, same thing. So again, you'll have to memorize these substrates in order. Have fun. So now we talk about the product. So in each step of the cycle, there's a possibility of getting products. So here we have each step that produces products and where they are in the cycle. So first, your acetyl-CoA, your 2-carbon compound, combines with a 4-carbon compound, your oxaloacetate. And that produces a 6-carbon compound called citrate. So again, 4 in oxaloacetate plus 2 in acetyl-CoA becomes 6 in citrate. So your citrate gets processed into isocitrate, and your isocitrate gets is the first one to have something ripped out of it. So your carbon dioxide, one of the carbons from isocitrate, gets ripped off, and in the process, there's free electrons to be taken up by NADH. So again, in this step number 2, it becomes a 5-carbon compound, alpha-ketoglutarate, and it produces 1 electron transporter and a waste product of carbon dioxide. Next, it gets ripped, another carbon atom gets ripped off again, and it becomes succinyl-CoA, and in the process, it creates another NADH electron transporter. Your succinyl-CoA can give enough energy to produce GTP, which is another different form of your ATP. So instead of adenosine triphosphate, we have a different molecule, but it functions similar to ATP. So 4th step, we have another energy molecule. Now in the 5th step, your succinate becomes fumarate, and in the process, it releases more electrons, but in this case, they're lesser energy. There's less electrons, there's less energy involved, so a different electron transporter takes these electrons. We have FADH2. Okay, so think of it as like NADH is the special Uber transport, while FADH2 is the cheapo tricycle transport. Okay, so after that, your fumarate becomes malate, and your malate has one more step where it can give, so it gives off its final high energy electrons to NADH and becomes oxaloacetate again. So in total, we have 6 NADH, we have 2 FADH2, and 2 ATP, again from 2 acetyl-CoA. So each one creates 3-1-1, so it became 6-2-2, since we have 2 acetyl-CoA molecules. So this is our net total from 1 glucose. Now we move on to our final stage in cellular respiration, the electron transport chain of your mitochondrion. So what happens here is ATP synthesis on the mitochondrial cristae. So again, your inner membrane of your mitochondrion. So this type of phosphorylation, or making ATP, is your oxidative phosphorylation, because again, we have your electron transporters and we're making them lose electrons. So we're oxidizing them. So your oxidized electron transporters fuel oxidative phosphorylation. And this is where most of the ATP of your respiration comes from. So again, your electron transporters come in and they pack a lot of energy in their electrons. So this is what happens. So your NADH and your FADH2 will give their electrons to the electron transport chain. So in some references, you might find names for these, ubiquinone or COQ. So these different enzymes have different names, but generally all of them are used to pump out hydrogen ions. So again, in the mitochondrion, your hydrogen ions are pumped out to the intermembrane space, where they will have a high concentration of hydrogen ions, which will then flow back into your inner membrane. So your diffusion of your high concentration of hydrogen ions will go back in, and that diffusion powers ATP synthase, which will be used to make ATP. Again, basically, you're using the exergonic fall of your electrons to pump out hydrogen, and then your chemiosmosis of hydrogen back into your mitochondrion will power ATP synthesis through ATP synthase, an enzyme. So basically, in oxidative phosphorylation, your NADH produces 3 ATP, while your FADH2 produces 2 ATP. So in general, we should get around 30 to 32 ATP for each glucose molecule in aerobic respiration. We call it aerobic because there is a final electron acceptor. In this case, in our case, oxygen accepts that final electron. So after all the energy has been taken from it, after it's been used up, what accepts it at the end is oxygen gas. And we need that so that your electrons don't just build up inside your mitochondrion. That's why we breathe, so that your oxygen can function as the acceptor of electrons from the electron transport chain, and it keeps your cell from imploding. Okay? So electrons need to be taken away at the end. Again, usually it's oxygen that's the final acceptor, and that's what we call aerobic respiration. So this is the most efficient form of respiration that we know of. However, there are other forms as well. We have anaerobic respiration, so this doesn't require oxygen. Some forms have other molecules that serve as the final acceptors, such as, for example, sulfide acceptors. So this is useful in oxygen-poor environments like hydrothermal vents. So at underwater volcanoes, the organisms that live there, since there's no oxygen, but there's lots of sulfide compounds, they use sulfide as a final acceptor. So it's less efficient, but it works. Another type of anaerobic respiration is fermentation. It's like a shortened version of respiration. So it doesn't go through all of the stages, only a short part of it. So it's less efficient, but also less dependent on electron acceptors. So first we have lactic acid fermentation. It's like glycolysis, the glycolysis stage, but instead of ending at pyruvate, your pyruvate is further reduced to lactic acid. So again, you can see this diagram here. It's basically your glucose being broken down to pyruvate. Same stages, energy investment and energy payoff. And then your pyruvate is reduced into lactate or lactic acid to produce what's this, to regenerate your NAD plus, which is used in your glycolysis part. Okay, so that's lactic acid fermentation. So yogurt is produced through such a method of fermentation. So you can make your own yogurt from milk and extra yogurt or even Yakult, and then controlling the temperature. Ethanol fermentation, on the other hand, is glycolysis, but instead your pyruvate is turned into ethanol and carbon dioxide. So you can see in this diagram here, again, glycolysis produces pyruvate, and then you remove carbon dioxide to produce acetaldehyde, and you regenerate your NAD plus by reducing that reducing that acetaldehyde into ethanol. So again, this produces an alcohol in the form of ethanol as well as carbon dioxide gas. So one example of ethanol fermentation is your bread. Your bread is made to rise using yeast, and yeast tends to do this kind of fermentation. So the carbon dioxide gas bubbles produce the air bubbles inside your bread. And another example is, of course, making alcoholic beverages. That's why a lot of alcoholic beverages have a fizz to them because of ethanol fermentation. There's carbon dioxide dissolved in your beverage. So photosynthesis, respiration, they're quite similar. One of them creates glucose, the other breaks it down. And this forms the carbon cycle of your living thing. So even plants, even photosynthetic organisms require respiration. So they break down the glucose they make in order to make more ATP. So again, these are two very important processes in the bioenergetics of life. So that's pretty much it for this session. I'll see you guys next time.
Party's over.